Okay, thanks. Yeah, I'm just giving an overview of the kind of data we are working with and some ideas of like how to process it in terms of like assembly and binning and so on.
Hello.
So this whole project is about biogas-producing microbial communities. And what we are doing is we generate metagenome, metatranscriptome, and single-cell data from these production scale biogas plants. And why are we interested in this? When you look at Germany, Germany decided that they want to cease the use of nuclear power by 2022. So they have to find some alternatives. And as of 2012, already like almost 12% of all the energy was actually coming from renewable energy sources in Germany. When you zoom in a little bit more into the renewable energy sources, you see that 64% of these sources are actually from biomass, and part of it is coming from biogas, mainly electricity and heat, actually. So these plants are used for mainly, I think, for generating electricity. So for those of you who don't have any idea how it looks like, so what you do is you take, usually in Germany, it's mainly maize, which is well, pre-processed a little bit, and that's put into these anaerobic digesters. And you also put some like cattle manure or other kind of like inoculum basically into the digesters. And then it's generating methane at the end. And the methane is burned and then used for electrical power or for heating up water or whatever, right? And then at the end, you can actually take the digestate and use it as a fertilizer. And so you have a sort of closed circle in the system. Okay, so biogas is, well, it's a clean and renewable energy resource. It has a favorable CO2 balance and so on. That's why we think it's a good idea to use it. But of course, there are limitations, especially in Germany, because the land is, well, we don't have enough land to grow enough biomass, actually. So we are competing with food sources. So that's why it's very important to actually understand the microbiology of these communities and then hopefully somehow, well, optimize this process once we understood what's going on. So how does the study look like? We have 4 different biogas plants. We look at the 16S structure. We do metagenomics, metatranscriptomics, and single cells. Let's look at the 16S first. What we have are replicates, like technical replicates for each of these 4 digesters. It's a little difficult to see here. And this is like the phylum level of the community structure. But you already see some differences between the digesters. Note the scale of the y-axis here. It starts with 50% here. So most of the microbes in here are actually firmicutes, like 85% in this case. And what you also see is the technical replicates look pretty similar, which is nice. And there are differences in the plants. And what we have are these 3 plants here are mesophilic plants, and the 4th one is a thermophilic plant. And what you see, well, it's really hard to see, but the big difference is basically, well, here you have these fusobacteria that only exist in this plant, and in the thermophilic plant we have the thermotogae, which basically don't exist in any of the other plants. So there are already big differences between the different plants.
It's quite a big difference between the technical replicates and frequencies.
Yeah, well, I don't know how much difference you would expect actually from.
So what are the replicates exactly?
It's a technical replicate. So they take one sample and then basically split the library construction and everything.
That's
right?
A lot of, I mean, it's like between 10% and 15% or something. I mean,
well, for some of them, you mean? Yeah, that's true. Well, but at least you can distinguish the different
just for
digesters, right?
Yeah, but just for technical replicate.
Could be the PCR. You have stochastic part in PCR, don't you?
Yeah, but you should have enough molecules that that's not a problem.
Yeah. Well, I don't really know what to expect actually, because I haven't seen many data sets which actually do replicates.
Yeah, I'm just
Right.
saying it's not.
So-
That's non-trivial. It's a long way.
Yeah. You might be right.
I mean,
Okay.
I mean, in practice, whenever you normally sample,
Mm-hmm.
normally what you're interested in, what's actually in the thing, and then you
Right.
sample twice. So there's even more variability. So this is greatly understating the actual variability, and still there's quite a lot.
Oh yeah, that's true. Well, if you take another, if you imagine this huge plant, right, and you sample from 2 different locations in this huge production scale thing, they might look completely different, right? So, yeah. Okay, anyway, so the point was we have 4 different plants and they have different conditions basically, right? And we also collect like the metadata of how much methane they produce and things like that. So the basic, the ultimate idea would be to somehow correlate this to the metadata. Okay, and then there's the metagenomics part. That's in collaboration with the JGI. What we have is, again, for each of the replicates, we have DNA and we get RNA sequenced. In total, we have like 340 this is actually gigabytes, so gigabases. So we have 400 gigabases for the DNA and 290 gigabases for the RNA, which we are going to assemble. And then these are some very crude metrics on the assembly. I just tried 2 different array assemblies here. And actually these are separate assemblies for each of the plants, right? And what you see is when you change the camera a little bit, I mean, of course the assembly looks different already. But we all know this. So we just picked one assembly basically. And what we also do is we actually combine the assemblies. So at the end, we combined all the samples because we think that we get higher coverage for very low abundant species, and in combining the data sets, we hope to assemble the low abundant species at least.
So would you expect to see the same organisms in the thermophilic system as the mesophilic system?
The
Presumably
same.
the
Well,
thermophilic
they
system
share.
is at 50 degrees.
I'm sorry.
Do you know what temperature the thermophilic system runs at?
I don't know exactly. I don't know. Maybe 60. I don't know exactly. Yeah. I mean, they definitely share some organisms, but there are also differences. Yeah. Well, the problem is I cannot really go into the details because we haven't looked at that closely. So that's just an overview what I present today. Okay, so what we did next is a combination of all these data sets and do one big co-assembly. And then, of course, we want to know which genes are actually present in which of the samples. So we map the reads back to the co-assembly, to the context of the co-assembly, and look at the coverage. Right? And what you see here, each data point is a contig. The size of the point is the size of the contig. So, well, you probably cannot see many differences here. And then the contigs are colored by their GC content, right? And this is comparing sample 1, like replicate 1 of sample 1 to replicate 2 of sample 1. And what you see is the coverage is, well, pretty similar between these two. But when you start comparing sample 1 to sample 2, you see a big difference here, right? But what you also see is that these clusters of same colors that show up, which could be maybe the same organism, right? So it's a sort of a binning of the context. So you can do this. You can of course compare all of these samples pairwise, and then, well, this is how it looks like for comparing sample 1 with sample 4. But when you look at this, you don't really know, well, is this spot here, this cluster here, is this the same as this one, or did it actually move here, or you don't really know. Of course, you can plot everything like at the same time, but still you lose the connection between the different clusters, basically. So we thought of just like doing some little animation, just to get an idea if these clusters actually stay together. And this is how it looks like. So what we do is we are morphing, basically. So this is the same here. This is In this case, well, let me stop it. Explain it first. I'll go back. Okay, so this is the coverage in sample 1. And now here we are morphing the coverage of sample 2 to sample 3, basically, right? And you have to, you basically see how the coverage is changing between these different, the different samples, right? And then when you watch the movie, we also do this for the x-axis. So this will be morphed from 1 to 2 and so on. So just to see how the difference look like. Right, and what you see is that these clusters actually stay together, most of them, which is, I think, another indication that these are actually really real genomic bins, right? And sometimes, if you look closely, sometimes these clusters get torn apart, but most of the time, it's that this whole thing just stays together, and then it's changing the coverage in one of the samples, basically. So here, it's basically, it has a coverage of like 100 in one sample and 0 in the other one, and so on. So it's really not, well, you get an impression of the whole thing, but of course it's not really automated, right? I mean, what you want to get out of it at the end is just the contigs as files, and then you can work with the contigs, and here it's just while looking at the data somehow.
Okay.
Mm-hmm.
Where is Chris?
Well,
Yeah, I was wondering that as well.
Doesn't he have an algorithm which exactly?
yes, and he has so Chris has an algorithm called concoct, and actually used the algorithm on this data set, right? So that's a snapshot again, and what we did is so what concoct does is basically it takes the contigs, it generates some chemo spectra, tries to cluster the contigs into bins using the chemo spectra, and it also uses the abundance differences between the different samples, right, to cluster it. So the next movie shows how these clusters look like. So what we did is basically from this snapshot, we just extract like one of these clusters, concoct, predicted, right? And what you will see is just one cluster, and then it's a movie which is showing different clusters, like each.
Did you also try the tool by Bielefeld, or?
I tried it; it didn't work. It just broke. So I have to talk to him to get it running.
Is Chris not here deliberately?
I'm sorry.
Does he know where he's supposed to be?
Of course, we'll send to everyone.
Yeah. Well, see here? It's a little bit fast, but anyway. So each of these snapshots, well, can you see that? It's really hard to see, but some of the clusters look pretty nice, but some of them are really like scattered all over the place, right? Let me try again. Oh, it's actually still running.
Can you turn down the lights?
Right. So what you want to see are these dense clusters, basically, right? And not clusters which are just scattered around, because what you wouldn't expect is a cluster where the coverage is somehow between like 1,000 and 10, because this obviously cannot be the same genome, right? Well, the assumption is that the coverage of a genome should be the same for all of the contexts.
It's not one genome, but you know there's variation.
Well, of course. Well, it could be multiple genomes which are very, very similar, like variants, for instance, right?
Yeah.
Which are very similar in terms of when you look at the chemo spectrum or something like this, and you might not be able to to distinguish them based on the chemo spectrum, but maybe on the on the coverage, right?
Do you think that this really means they're false in some sense?
Well, I wouldn't say they're. Well, I don't know. I don't really know. I mean, at least what you see is that the colors are the same always, right? So some of the clusters, it's not like randomly putting together genomes with different GC content. The colors are GC content, right? I think it would be a problem if you see like red and blue dots on the same picture, right? As before. So here the colors are always the same. So the contigs seem to have some sort of similarity, but the coverage of a context is still very different. And
Sorry, so can you explain to me, because I'm really slow at this,
Yeah.
exactly what the axes are? So I understand that.
That's a
we're
okay,
doing
let me
two
go back.
different samples, but
Yeah.
what are the numbers?
okay, so this is a snapshot here. This is comparing. So each dot, again, that's this one here. That's a pretty big one. It's like 100 kb contig here.
Okay.
Yeah?
Yeah.
Each dot is a contig, and this is showing the coverage of this contig in sample number 1 versus the coverage of this contig in sample number 2. So in sample number 1, this has a coverage of like, what is it? I don't know, 200. And this is and here it has a coverage of 10 only, right?
Okay, so it's more abundant in sample 1 than it is in sample 2.
Yes. Right. Okay, and a binning tool should pick up something where the coverage is actually the same, right? At least in one sample. Right? Because the genome has the same coverage in this one sample.
Right. Right.
So it should be a dense sort of cluster.
So what can happen is if you don't have a background which has the same, if you imagine a situation with variation, you have a genome which has the same coverage everywhere, but then there are variants which have different coverage in different pools. Okay, so maybe what his program is doing is it's lumping the variants because they overlap with the bits of the genome which have even coverage, or it's putting them all together.
Probably, yeah. Well, I'm not really sure. I have to talk to him.
That's why it's
Yeah.
disappointing.
Well, I'm going to talk to him. Yeah.
I want to see how group M performs.
Well, I don't have any results. It didn't work. It just broke.
So these are metagenomic reads, right? They're just
Yes.
shotgun sequencing.
Yes.
Okay. So there's potentially another issue, which is presumably, because you're sequencing very deeply, right?
Uh-huh.
The different DNA molecules can potentially break at different places.
Mhm.
You know, particularly if you've got transcription-heavy sites versus, almost untranscribed sites. So there's also a chance that you're getting some bias because of the underlying biology of what's being expressed and what isn't, even though you're looking at the DNA level,
Okay.
I think.
Well, but the problem we are facing is we want to bin the contigs into bins,
Yeah,
right?
No, absolutely.
And the information we have is the coverage of a contig and like the Kmer spectrum, right? And similar Kmers, like a similar Kmer spectrum, should bin in, and then also a similar coverage should bin it, right? Very closely related genomes have very similar Kmer spectrum, but they might have very different coverages, right? And the idea of these binning algorithms is that they actually tear it apart, right? Okay. And
Did you use a different assembler? What assembler did you use?
right,
Oh, the same assembler. as-
this is the same assembly,
Ah,
working with the same assembly.
yeah, like that connect method was also developed for Ray, right? Or with Ray?
It doesn't matter, any kind of assembler.
Yeah, but the
Well, you
correct
assemble it
assembler
first, you
is
map
at
the
least
reads
with-
back, and then
yeah.
that's how it works. Yeah. Right. So I don't know. We have to look more closely what's going on. And I remember Chris saying it's actually only working for like, I don't know, 40 samples or something. Maybe that's the issue. I don't know. Anyway. Okay. So the next data I just have a glimpse into the data here. So we also do metatranscriptomics to see which genes are actually active, or let's say transcribed. So what you see now is that, again, this is, so now you're looking at genes, right? So what we do, after we have the assembly, we run a gene prediction tool, and now we are looking at genes here. So each point here, each data point here is a gene now. And we are plotting the DNA coverage of a gene against the RNA coverage of a gene. So we map the DNA reads back, and we map the RNA reads back, and now we compare the coverage, right? And yeah, well, one of the questions we have is, which I cannot really answer, is if you want to compare the samples, right? You can look. You are interested in differently expressed genes, basically, in the different samples, right? But at the same time, the coverage of the genome, the abundance of the genomes might change, right? So you can say, okay, I don't care about the genome, the abundance of the genes, I only care about the most highly expressed genes, so I just compare one sample against the other just by looking at the RNA counts or something, right? But here the question was, well, if a sample, or if a genome has very high coverage in DNA, does it also have or is it somehow related to the coverage in the RNA? And then also, what kind of genes are actually expressed? So we did an annotation of the genes and a taxonomic classification of the genes. And what you see here are these blue dots now. These are, this is the, what is this, a family? I don't remember. Methanomicrobia. So these are genes classified as methanomicrobia. And you see that there are obviously genomes from this clade which are different, have different coverages, right? But we can also look at the genes. And here the red dots are genes that are part of the methanogenesis pathway, basically. So this is the MCR genes. And what you see is that these genes are really highly expressed in well, at least in this sample. Right? And it's also somehow correlated to the DNA coverage, which means there are lots of methanogens, actually, pretty high coverage, which also produce a lot of RNA, this particular gene. Right. But there are also some which have a lower coverage here, much lower coverage, DNA coverage, and they almost produce the same amount of RNA, basically. So higher activity, maybe. Okay, and then we also do some metaproteome analysis. This is a 2D gel of the same sample, and it's nice to see that you can actually relate the metaproteome analysis to the transcriptome analysis. But what you have to do is you have to use the assembly as basically the database for the metaproteome analysis, right? Because if you try to identify one of these spots, or identify the spots and then classify the spots, if you use a SwissProt database, only like 26% of the spots can be identified. If you use our assembly, it goes up to like almost 70%. And that's because the genes don't exist in any of the public databases. And when you look at the annotation, again, you see these genes which are part of the methanogenesis pathway. Okay, and so what time is it? 2 more minutes. Okay, I just want to spend like 2 more minutes maybe on the single cell. Or 5 minutes, yeah. Okay. So that will be the next step. So we also did some cell sorting to get single cell genomes. Actually, the cell sorting was done at the JGI again. So what we have like 768 single cells sorted. What they do is they sequence, well, they lyse the cells, they amplify do an MDA, a round of MDA to amplify the genome, and then they sequence the 16S gene to actually, well, identify the genomes, basically. And that worked for like 120 of these, because not all of them get lysed or whatever, and maybe they didn't catch the 16S gene or whatever. So we will get 120 or 100 single cell genomes, basically, from one of these digesters. And so the kind of workflow I developed when I was still at the JGI is like an assembly and QC workflow. The assembly workflow is sort of, well, outdated because Spades became available, and turned out that Spades is actually doing much better. Well, I wouldn't say much better, but it's doing a little bit better. But you have to be very careful about contamination, of course, in these single cells. So we have this QC pipeline where you basically try to, again, taxonomically classify your reads and your contigs, and you can do some like analysis on the contigs to identify possible contamination. And what we do here is a t-SNE analysis. So each of these little worms here, this is basically one contig, and you're looking at a sliding window across the contig, right? So each dot here is one window. You're sliding the window across the contig, so that means you get multiple dots here for just one contig, right? And they line up pretty nicely in these kind of like worm structures. And then the t-SNE is a dimension, how do you call this, dimensionality reduction method. Other people would probably use like PCA or something, but this is much nicer to visualize because it's much better suited to do the visualization in like 2D, right? Because it's always trying to reduce it to like a 2D space. And what you want to see is just one cluster. Oh, okay, and I have to explain what you're looking at. So we take this window, we compute the Kmer frequencies, again, tetranucleotide frequencies, and then we are comparing these windows against each other. And what you want to see is that everything clusters together, basically. And what you don't want to see is something like this, because that means there are contexts with very different tetranucleotide frequencies.
Sorry, one question. What does the color tell?
The color is just the contig, so that's easier to identify. So that you can, that you see that these spots here, all of these are from one contig and these are from another contig. It's just different contigs are just random colored, basically, right? But whenever you see these dots next to each other and having the same color, it means they come from the same contig, basically, right? So that's a very powerful way of detecting the contamination if the contigs are large enough, of course, right? Because if you have just small contigs and you have only one window, you might not even see it. And you might not be sure if it's actually this one, for instance, could just belong to this one here. And it's not really contamination, but like overall, I would say that here, these are two genomes in here and same here. Okay, and then we did some benchmarks on the single cell assembly. I will just skip this. I just want to show the last thing we are working on. Oh, this is actually the benchmark on our assembly pipeline. This is our assembly pipeline down here. This is Spades. This is an early version of Spades. So you can probably just look at like a few of these columns. So this is a total assembly size. Each color is a different species. So what we have, let's go back. What we have are these single cells from these organisms, like E. coli, M. ruber, P. What was this? Hipparium. And then we have 8 single cells for each of them, right? And then we measure the total assembly size for the different assemblies, and we come up with some sort of gold standard. The gold standard would be a region of the genome. So when we map the single cell reads back to our reference genomes, we can compute the coverage, and if the coverage is above 5x, we keep this region, and this is what's left afterwards. And this is, so to say, what a very good assembler would be able to assemble, right? And you can see that there is a big variation between the different cells, right? Because you only pick up like 50% of a genome using the single cell sequencing. And when you look at Velvet, the total assembly is, well, it's a little bit worse, but when you look at the number of contigs, then it's really bad, right? So the gold standard would tell you, okay, the number of contigs is between 1 and 520 in this case, right? But no, actually, that's 520. So this is the number for Velvet, actually. So Velvet produces many, many, many more contigs, right? It's just totally fragmented assembly. Many contigs, and they're very small. So this is the N50, and the largest contig you see compared to the gold standard, it's very fragmented kind of assembly. There's a special single cell assembler, Velvet SC, which is even worse than Velvet, because the total assembly size is just tiny, and it basically has the same problem. And then we also use IDBA, which looks pretty good actually in terms of total assembly size, but number of contigs is pretty bad. Again, very fragmented assembly. And now you see that the early version of Spades is doing pretty good here, and it's comparable to our pipeline. And what we did in our pipeline is the problem for the assembly is when you look at the coverage of the single cell sequencing, the coverage is highly biased, right? So you have regions with only 5x coverage, and you have regions that have like 15,000x coverage, right? And that's because of the amplification step, right? The MDA, multiple displacement amplification. And that's a pain for the assemblers. The assemblers cannot deal with it. So what we do is we normalize the coverage, basically, computational, computationally to something like 50x coverage, and when you do the assembly afterwards, it looks much better, right? And then we do an assembly using, I think, all paths in this case. And now it looks much better and much closer to the gold standard, actually. But these two are actually very, very similar, I think. The number of mismatches is pretty high with Spades, but as I said, this is an early version of Spades. I think it's probably better now. Yeah, okay. Okay, and then we also do co-assemblies of single cells and metagenome, because as I said, you only pick up like 50% of your genome. When you look at one single cell, and this is the percent of genome covered by the assembly, when you're looking at only one cell, well, this is actually a very nice example, because the coverage was between like 65 and something like, I don't know, 98% of a genome. But when you look at this guy here, we only recovered between 25 and in best case 70% if you just take one cell. But when you start combining the cells, the different cells, right, it gets much better. So these numbers here are for combining the data sets of two different single cells. You see that the coverage gets higher, and when you end with a coverage, well, with like, I don't know, 4 single cells, you're already at like 80% of a genome. So if you have multiple single cells available, you can actually reconstruct more of a genome. But we thought, okay, we also have a matching metagenome available. So why not use the metagenome to actually reconstruct the rest of the genome, right? And that's what we do. We take our single cell reads, and then we try to reach into the metagenome reads and pull out the metagenomic reads, which are part of this genome, right? And that's the result. So here you see a single cell assembly up here. It's pretty like fragmented. These are the single cell reads, again, with the bias and the coverage. But when we take these reads here and then match these reads against the metagenome reads, and you look at the recruited metagenome reads, the coverage is much more even. And if you assemble these reads, you get just one contig from this example. And that's working even better for regions which are not assembled at all, not covered by a single cell assembly. So this is nothing up here, but we still have some single cell reads here. And of course, the coverage is so tiny that you cannot even assemble it, right? But when we take these reads and then pull out the metagenome reads, which match to these reads, we get a better coverage, and then we can assemble these proxy reads, and we get an assembly of this region, which we didn't get in the first place. But of course, again, we have to make sure that we don't like pull in contamination of the metagenome and so on. So the whole thing has to go through a QC pipeline again.
Do you have paired?
I'm sorry?
Do you have paired, long paired ends?
No, usually they are short paired ends, but we can do this like iteratively, and then we pull out even more. Yeah.
So that, yeah, it'll tell you pretty quickly if Chris's method is working. I mean, once I have a single cells. Yes, that's true. Or we're still waiting for the data. Yeah. Yeah. Okay. Yeah, that's it. So Andreas Bremges is a PhD student in my group. He's a worker on these co-assemblies. These are the guys generating the data, the biologists. The clustering you've seen, like the t-SNE analysis, that's done in Barbara's group, and of course JGI is sequencing and doing all the single cell stuff. Okay, that's it. Thanks.
I think we have time for 1 or 2 more questions.
I can keep going. So you could also put the single cells into Chris's method as well as another way of doing the same thing. So
That's true, yeah.
that would
Actually, that's a good idea. I mean, we don't have data for these samples, but we have lots of single cell data. But you have to
so just put them in as
put them in,
they
but
are a metagenome. They are a metagenome. Just treat them as a metagenome.
yeah. Well, but we don't have different coverages. That's the problem. His tool needs these differential coverages.
Together with.
Oh, together with the other data. Yeah, that's true, actually. And then see if they show up at the same place, basically.
They should.
Yeah.
They should just be informed. They're very informative.
That's true. Yeah. Yeah.
Okay, well then, Alex again.
I have a question.
Oh, okay.
Actually, even two. One was about when you checked the contamination of the single cell assembly, you did it based on k-mesh, right? So,
Yeah.
But in the slide before, there was a picture with this paired end, such I think I saw these such plots in talks from Matt Albertson, and they recruited. He used paired end information to recruit additional context.
No, we don't do this.
No.
No.
Okay, so the idea is whether you get one consistent.
Just one consistent cluster.
Yeah,
But
but you
you
can
just based on the tetragonal frequency right now.
Okay, and did you think about changing the binding method from compound to something else?
Well, of course, you want to try GroupM, for instance. There are several methods. Like, as I said, GroupM didn't work because of some software problems. So, how to talk to
Right.
what's his name again?
Michael Mofford. Yeah.
Yeah.
Okay.
And Max being about methods. We have tried.
Okay. Well, thanks.